so we're going to go over the topics of DNA that we talked about. We looked at the structure of the DNA, replication, central dogma, DNA versus RNA, and then protein synthesis, which will deal with transcription and translation. So first, sometimes on the keystone they'll talk about the structure of DNA, and we want to know that it has a double helix to it. If we look here, we can see that these are the major things of the structure of DNA. The first being that it's a double helix. It has the double strands, which is important, and that's another way to differentiate it between RNA and DNA, is the DNA is going to be double-stranded. It has the nucleotide building blocks, and it's going to have the sugar and phosphate backbone, and then it's going to have the nitrogen bases in the middle. The bases we have are adenine and thymine, and then guanine and cytosine, and remember, they're always going to be equal to each other. So if you have 46 guanines, you're going to have 46 cytosines. If you have 34 adenines, you're going to have 34 thymines. It's always going to be equal. A will always be equal to T, and G will always be equal to C. A way to remember which base goes with which base is you can think apples in a tree, A goes with T, and a car goes in a garage, C goes with G. Just an easy way to remember those two matches. Again, complementary base pairing, they complement each other. A always complements T, G always complements C. And then we also have the backbone. When you look at the backbone, you're going to have your phosphate, sugar, and they're going to alternate the whole way down the backbone. And every time you have one of those sugars, it's going to connect to the nitrogen bases in the center. And when you're looking at the DNA strand, you always remember the nitrogen bases look like the rungs of the ladder, and then the sugar phosphate are those outside ledges. And then in the middle, when we look at our hydrogen bonds in the middle, they're going to connect the nitrogen bases. On the outside, you're going to have the covalent bonds. And if you remember from the beginning of the year, we learned covalent bonds are stronger than hydrogen bonds. And we need hydrogen bonds in the center so that DNA can be replicated and we can break those bonds with DNA helicase. So the weaker bonds are always going to be in the center because that's where we break it, right down the middle. Then on the outside, where the sugar phosphate, those are going to be the covalent bonds. And then the sequence of the bases is what gives us our genetic code. So as you look at the sequence, if it's A, T, C, G, that'll give you one genetic code, or it can be G, C, C, A, whatever it is, that tells you what you're looking at. Then we talked about the idea of the central dogma, and this is just the idea of DNA, going to RNA, and going to protein. It's that overall process of how DNA codes for our proteins. The first one, DNA to DNA. So you're making a copy of DNA. This is called replication, and this can only happen in the nucleus. You can remember this because DNA is double-stranded and it's too big to go outside of the nucleus. So anything that has to deal with DNA is going to have to take place in the nucleus. The second thing that will happen is DNA will make RNA. When DNA makes RNA, again, you're using DNA, so it has to take place in the nucleus. And we're going to call this transcription. We're transcribing RNA from DNA. So DNA will take the one strand and copy it into RNA. When you do this, the base pairs will change from thymine to uracil in the RNA. Again, RNA is different from DNA because it's single-stranded, and then since it's single-stranded, it's able to leave the nucleus. When it leaves the nucleus, it's going to travel to the ribosomes where translation is going to take place. And this is where we're going to make our proteins, and this is in the cytoplasm. So then the RNA codons code for the amino acids that will join together to make the proteins. And that's all going to take place at the ribosome.
Can I just jump in here for one second? Okay, this whole idea of the central dogma is like the big idea of how we make you. We start as a code and then we end up as a person. We are made of proteins. When you look at each other, you're simply proteins. So in my class, I had a little action to help us remember the central dogma. Did you do this?
No.
Okay. So it's DNA to DNA to RNA to protein. So DNA is double-stranded. So it's DNA to DNA to RNA to protein. And you point to yourself because you're made of protein. Okay, DNA to RNA, single-stranded, to protein. And that's the central dogma.
I have one more thing to quickly add, an acronym that we use in my class. We call it Dr. CAP. Okay, DNA, RNA, codons, to amino acids, to proteins. Just that way you can remember the 5 steps. Think Dr. CAP. So DNA goes through transcription to make RNA. We chunk it to make codons. They're translated to make amino acids, and they join together for protein.
The action though.